Hi, everyone. So nice to see you all. I mean, I'm really happy to have such a wonderful audience for something that I'm sure is going to be a very fruitful discussion. I'm sure many of you have seen in the news terms such as CRISPR-Cas9 and the famous Chinese twins that everybody's nodding their head, that has actually spurred broad conversation in the United States and beyond. But the conversation didn't start with just that news item. As a matter of fact, the idea that we could precisely edit the genome is not new. We have started with the GMOs in the early 90s, actually, as applied to crops and other plants. However, the fact that we can move now toward the human genome and potentially edit very precisely for traits that are important to us is something that actually generates a vast array of questions that we hadn't really thought through before. And to discuss this question in general and more precisely as they relate to that news item I was discussing a few minutes earlier, we really have a panel of wonderful people. I have to say that we're very fortunate at UW-Madison to have such an expertise to tackle these kind of issues and to encourage what we think is extremely important for issues that are not only scientific, but also ethical, legal, and have societal implications that all of us, we should think about together. It's not the scientists that can answer those questions, it's really all of us in society, and I think I really want to thank, I don't know where she is, over there, Reed and Laura here for giving us this opportunity. So we have asked the speakers to each give a 5-minute, very brief presentation to set the stage, and then we're going to move towards a moderated discussion before opening the floor for discussion with the audience. And we are going to start with Kris Saha, and I'm going to ask Kris to introduce himself. And we have asked Kris to actually tell us what is gene editing generally, and CRISPR's technology in particular, and what is the current state of the science and its application. And he's going to do all of that in 5 minutes. Yay for Kris. Kris? Come on.
First of all, thank you everyone for coming, and also Dominique, Wid, and Laura for organizing this event. So 5 minutes, here we go. I think one of these minutes will be changing PowerPoint. Okay. All right, so my lab works on the 4th floor here. We work with a number of scientists across the campus, clinicians, biologists, material scientists, to think about genome editing, and I'll try to march you through a few fundamental concepts here. Genome editing is the idea of directly changing the DNA code. So here at the top, we have a single base pair change. Very light, but it's highlighted in T there, between the genome of a healthy individual and one that would have a sickle cell trait. And down here, these are also these different symbols along this line, which is representing a gene, are the different other types of base changes that you would find in patients, in this case, a small child with immune deficiencies. And so the idea that we can go ahead and directly flip this T back to an A in patients' tissues, as well as perhaps their blood system, is one of the ideas behind genome editing. How does this get done? This is a video from a group in Germany that talks about various components. So this orange molecule here represents an RNA molecule. This purple blob is a protein called Cas9. And over here, this blue molecule is a DNA. And so what the CRISPR system, one type of genome editor, does is it loads the RNA within the protein. And once it's inside the cell, it scans the genome for these types of typographical errors. And so this error here in red, once it is near this area and it matches the sequence in the RNA, you see that the protein ends up cutting the two blue strands, which is the genome. Once the genome is cut, the cell takes over and it tries to repair this cut end. And if there is this third template, this DNA strand, it will be used as a template to put in whatever sequence was in this strand. So we can control the sequence of this blue DNA molecule that we put in. We can control also the sequence of that orange molecule. And this is how we get precision in the CRISPR system. A lot of things are simplified in this cartoon, right? So it's not easy to put everything in. It doesn't only make cuts at that red site. It can cut elsewhere. And so these are some of the challenges with this technology. Regardless, there's a number of groups, namely one at the NIH, that's attempting to develop better genome editors. So I co-lead this NIH Somatic Cell Genome Editing Consortium. And the idea here is that we could deliver these genome editors to various tissues, say the brain, for instance, specifically inside patients. And one of the challenges here is to get this protein RNA DNA complex into the right cells at the right time, avoiding other tissues, importantly reproductive organs, such that you edit cells, the genome in the cells that you want. We're also interested in bringing new types of machinery to that specific place in the genome where there may be a typographical error. And so that's shown here. These types of complexes and proteins could be modified. And so this is really a nationwide effort that's coordinated through the NIH. We have different viruses, different polymers that are delivering these things. And we're also making microtissues within chips using human cells, some human embryonic stem cells, and pluripotent stem cells that are developed here to test these genome editors before they go into people. And so what can this technology do? I think that video highlighted applications of direct injection into patients as therapies. So you go to a clinic and they prescribe you a genome editing therapy. That's certainly one application that there's a significant financial interest in. There's also a number of applications as research tools. So in drug screening, it's extraordinarily useful. Almost every pharma company now has a CRISPR-like or genome editing division. It can be used in toxicology to figure out how toxins work. And it's really a tool that's being used throughout campus by fundamental biologists, basic biologists as well. And I've, at the bottom here, highlighted a number of products that really have come on board and approved by the FDA over the last few years, focused on gene therapy. None of these down here are specifically using genome editors, but the anticipation is that these products will be involving genome editing in the very near future. And so some of these products, perhaps you've seen in the New York Times, called the CAR T therapy, where you can engineer your own T cells to go after cancer. That type of genomic engineering could be done with these genome editing tools. And so the actual clinical trials underway right now are shown in this table. I actually had to split it up into two halves. This is a review by Matt Porteous out at Stanford, who put this together very recently. And you can see there's activity from 2009, notably from a company called Sangamo that developed an earlier version of genome editors called zinc fingers that have already gone through extensive testing, and that all the way to new trials, in this case 2018, and there's certainly new ones that we could add for 2019. A lot of these are in the blood, some are in the liver. All of the tissues that people are excited about really span the entire body. And so my final slide here is to kind of highlight what here at Wisconsin we're doing. Certainly in terms of somatic editing applications, we have a number of new transgenic animal models that we're trying to develop using CRISPR, perhaps to inform preclinical studies. We're using some of the stem cell engineering expertise we have on campus to test new types of genome editors. We have material science projects to allow these genome editors to, say, get into the brain more effectively and edit the right part of the genome. And some of the disease targets here are focused on the eye, the muscle, newborn screening disorders, Alzheimer's disease, neurodevelopmental disorders, and many, many more. I'm sure that's not captured on this slide. So hopefully that gives you a snapshot in 5 minutes of what we're trying to do for somatic editing in particular, and you'll hear a lot more in this panel. Thank you.
Thank you, Kris. We're going to move to Jeff Jones here, who's going to be talking more particularly of the opportunities of making changes that can be inherited to future generations. And you're going to also explain what we mean by that. So give me 1 second here.
Okay, so I'm the lab director at a fertility clinic in town and also work in the Department of Obstetrics and Gynecology. So I want to talk about some of the controversy that we're going to be talking about tonight is editing the genome of an embryo. And that way, any genes that you edit, they're inherited by future generations. So there's a lot of controversy about that. Should we do it? But what I want to do tonight is just briefly show you the procedures that we use in our lab every day, and then I can apply this gene editing technology to that. So in vitro fertilization, basically what we do is, so this is just a reproductive tract of a female with an ovary. We stimulated a woman to produce more than one egg, collect those eggs, put it into a petri dish, add sperm to it, let them develop from 3 to 7 days, and then transfer those embryos back into the uterus to produce a baby. So since we have these eggs and embryos in a dish, it gives us an opportunity to introduce or edit the genome of these. One of the techniques that we use, rather than just putting sperm and eggs in a dish to fertilize embryos, is to use a technique called intracytoplasmic sperm injection, or ICSI. And we do this using an inverted microscope and micromanipulators. So as you can see on this, we have 2 holding pipettes. One holds a mature egg here, and you can see it's mature because it has a polar body at the top. And then we take another pipette in which we can pick up a sperm and inject it into the egg rather than letting the sperm itself fertilize the egg. So this is called intracytoplasmic sperm injection. And I'll just show you a little video of how this is done. So we got the egg, got the sperm in the pipette there, stick it into the egg, pull out the cytoplasm, and then expel the sperm with that cytoplasm back into the egg. So these eggs are very tolerant of this procedure. We've been doing this for about 15 years now. Fertilization rates are exactly the same, if not better, than conventional insemination IVF. Embryos develop the same. It's a very well-established technology. But by doing this, we have the ability then to use CRISPR to edit the gene in this single, still an egg, into an embryo. So that when it develops, every cell contains that edited, the gene that was edited. So it's consistent. You don't have just part of the cells of the embryo containing this edited gene. So this is this is the stage that the embryo is at when we transfer it to the uterus. It's called a blastocyst. So just like a chicken egg has a shell, the human embryo also has a shell around the outside. It's called the zona pellucida. It has an inner cell mass. This actually becomes the baby, and then a group of cells called the trophectoderm that become the placenta, the thing that attaches the uterus and nourishes the baby during development. So an advantage of this is we can actually sample these cells and test whether this gene editing worked by taking some of these cells and doing genetic analysis of them. And that is called pre-implantation genetic testing. And this is a video of that process. Thanks. But the video is not going to be working. Okay, so we're holding that embryo, and then we use a laser to cut a small hole in the shell around the embryo, and then use that laser as we're pulling out some of these cells to cut that off in a sampling tube. Again, every IVF lab in the United States does this every day, very common procedure. We usually do this for patients that are of advanced maternal age. Maybe they're, don't want to avoid having children with trisomy 21 Down syndrome, so we can send these samples off and tell whether the editing happened. So that's how this technology could be, I don't want to say simply applied, but it's not a large step. There's not a lot of new technology in the lab that we need to learn to do this in human embryos. Thanks.
So as Kris and Jeff alluded here, there's a lot of if, potential, and so on. There's a lot of questions that are unanswered. And more particularly, there are a lot of questions that actually are not just scientific, but that resolve around ethics. And Pilar will actually tell us what are the main ethical questions raised by the technology.
Hi, I'm Pilar Ossorio. I'm a professor of law and bioethics at UW and also the ethics scholar in residence at the Morgridge Institute for Research, which is also a part of this building. And so what I'm going to do is just raise some issues. I'm not going to analyze them. I'm not going to talk about how they might be answered. These are just concerns and ethical issues that people have thought of over the years. And I would first have to say that although gene editing in humans is a newer idea, the idea of making genetic modifications to human beings is not a new idea. We began doing so-called gene therapy in the 1990s, and so ethicists have been thinking about the ideas, thinking about the issues related to making genetic modifications of humans for a long time. And I would also start by saying that in the United States, for medical uses of gene editing, there's a substantial amount of oversight and regulation that would apply, both to the research and to the sort of further downstream attempts to create a medical use. And we can talk about exactly how that works later. So you heard Kris use the term somatic. So I just want to make a distinction really quickly that when we talk about somatic cell editing, we're talking about edits that would affect the person who was edited, but they wouldn't be passed down, they wouldn't be inherited. So I'm going to use the term somatic cell editing and then contrast that to inheritable editing or inheritable modifications. And I try to steer away from even the term therapy because not all uses might be therapeutic in the way that we typically think. So what are some of the issues? First of all, there's a whole suite of issues that might apply to either somatic cell editing or germline editing that are pretty, in some ways, standard research ethics issues. If you're going to do these things, first you have to study them. You have to try and develop them in a way that they will be safe, in a way that they will be effective in achieving some goal. So to study them, well, we have to ask questions like, how much evidence of safety do we need before we would try this in a person the first time, right? So that's a kind of typical ethical issue that has to do with the research. As I said, and that could apply whether we're talking about inheritable modifications or whether we're talking about somatic cell modifications. And as I said, there's actually a pretty well-developed set of ethical approaches to at least address this question. It's not a kind of question, and the research ethics questions generally are not that distinctive for gene editing. We've had to address these questions in other contexts before. I think when we get to inheritable modifications, those we sort of haven't addressed so much before, at least not intentionally. Most of the kind of scientific research that we're doing in the medical realm, we're not intentionally trying to affect the next generations. And so that raises a lot of questions about how would you sort of detect the long-term effects that might happen 1 or 2 generations down the line. How do you keep some kind of observation of offspring of the person that you modified? How do you do that in a way that respects that person's autonomy? After all, they never volunteered to be your research subject, right? So that does raise, doing it inheritably does raise a set of questions that we haven't really handled before in research ethics, but at least we have infrastructures. We have research oversight law, we have committees, we at least have the infrastructure to start to handle those kinds of questions. When you talk about inheritable modifications, you're also talking about a whole other set of issues that people raise. So one of the comments that I've heard a lot is that, but those people who are going to be born in the future, they didn't consent to have you mess around with their genome, right? So that's something that worries a number of people. I think we have ways of addressing that, but, and frankly, people who are born in the future don't consent to anything we do that might affect them, and there are a lot of things that we do that might affect them, right? But part of what that means is that because they don't really have any choice in this, it does perhaps raise the bar for us with respect to really trying to minimize risks and potential harms and ensure that what we're doing is being done only for very good reasons, right? We need the strongest of justification to try and do it if we try and do it, and we really have to work to minimize the harm. There are other concerns like the idea that we could somehow change the human gene pool, so change our sort of collective genetics in a way that might be bad. People are concerned that we might render children we might turn them into a little bit like things, right? That they would be subject to their parents' will in an inappropriate way, that they would be designed or constructed in ways that parents shouldn't treat their children. And I think we have a whole set of questions that have to do with justice and fairness. Who will have access to these technologies? Will we be creating will we be sort of biologizing social inequality in a way that we hadn't done before? So with that, I'm going to stop and turn it over to my colleague Alta Charo, who will talk to you specifically about issues relating to the Chinese work.
Thank you, Pilar. Yeah, so Alta, actually, I believe you were in China when that announcement was made. Alta was actually at that conference where the Chinese researchers claimed that he had edited the genome of baby twins. So you can tell us what exactly happened and what's the global response and what it means for the ethics related to that research.
First, thank you. Hi, I'm Alta Charo. I'm a professor of law and work largely on regulatory policy and ethics issues related to biotechnology. So for those that are going to throw their minds back to Thanksgiving weekend of November, you may have noticed that there was coverage in the newspapers of a Chinese researcher named Hu Zhonghui, commonly referred to as JK. That's the kind of nickname he uses. And he had, for the first time, announced publicly that he had used the same CRISPR technology that Kris has just described in order to edit embryos, and did not stop at pure laboratory work in order to understand how this functions, how to make it specific and precise, how to avoid off-target edits that might have unwanted effects, but instead had proceeded, once he had edited these embryos, to then implant them, and had now brought them to term. And 2 weeks prior to Thanksgiving weekend, he says 2 girls were born, twin girls, fraternal twins, who had edited genomes. It sent shockwaves through the entire scientific community globally. Partly it was because there had been hints that he was interested in doing this for quite a long time, conferences going back several years, where he talked about this and where he was regularly told this is not a good idea. They were also shocked because prior to this moment, there had already been some significant events that had outlined some of the reasons why we shouldn't try this at least now. There had been a major summit in 2015 that was organized by the Chinese Academy of Sciences, the Royal Society from the United Kingdom, and the U.S. National Academies of Science and Medicine, and brought together about 750 people to discuss CRISPR-Cas9, and had specifically said that we don't have the science basis yet, among many other things, to even think about trying this in a clinical context. The safety issues were overwhelming. Public discussion had not yet really developed. Subsequent to that, there was a long-term consensus study that was international in scope. Dietram Scheufele, who's here with us tonight, was a member of the committee. I co-chaired that committee for the National Academies. And it came out with a report that to this day is still the only really comprehensive set of kind of an ethics code and a scientific code for whether and when one should proceed with this technology, both somatic and germline, or heritable, as Pilar has been discussing it. And it specifically said, number one, you need to have enough scientific evidence that it actually is safe and effective. We're not even close. It also said it should never be done except for the most compelling purposes in order to avoid truly serious, devastating diseases that can't be avoided otherwise. And there are many other ways you might be able to try to avoid diseases. Jeff talked about being able to test embryos for mutations. And so for those people that are willing to do this, you can simply not implant an embryo that shows a really terrible mutation, and instead use an embryo that looks like it's more typical. So there's really very few instances in which our committee could isolate circumstances where there might be no real alternatives because things like that kind of pre-implantation diagnosis wouldn't work. We also talked in that about the necessity to have a protocol that not only had gotten lots of review from independent eyes, so you don't get overly enthusiastic about your own science, but that you have a game plan for what Pilar was mentioning, which is long-term follow-up on the health of those children and of their children. And that might be something we can't even figure out how to do. That multi-generational follow-up may turn out to be an impossible condition. And yet we thought it was important if we were ever going to do this in human beings. So we thought with that report that we had actually laid out an incredibly rigorous set of conditions before anybody would try this for heritable purposes, and instead focus on the more important somatic purposes, some of which were hinted at when Kris mentioned sickle cell in one of his first slides. Sickle cell, muscular dystrophy, certain forms of blindness are already major targets. But instead, J.K., He Jiankui, had developed a kind of fixation on using this not for any of these compelling purposes, but instead for something that actually didn't make much sense at all. It is true that in China there is a high rate of HIV infection and also an extraordinarily strong stigma that really disadvantages people who are infected and disadvantages members of their families regardless of whether they're infected. So what he did is he recruited couples where the father was positive for HIV and said to them, what I'm going to do is I'm going to edit the embryos of your children and I'm going to make them resistant to HIV infection. This made no sense at all. First of all, if these children were going to be infected by their parents, it would be through the mother, through the course of the pregnancy, and there are ways to actually reduce to extremely low levels the risk that the father's HIV status would affect the children. Second, it's a ludicrous way to try to deal with the problem of HIV infection writ broad because you can only do this for a few people. But mostly, he didn't have enough scientific expertise to actually achieve what he wanted to achieve. There is, in fact, a mutation, and if you were to throw up that slide that you had from Matt Porteous of all the different clinical trials, you'd see it. On that slide, you'd notice there were a number of trials that concerned something called CCR5. This is a particular gene sequence that has some variants which will make somebody resistant to HIV infection, and it is predominantly found in Northern European populations where it is found at all. So there are somatic trials now going on to see whether or not we can use that information for people who are already born. This would not be multi-generational. He thought, well, I'm going to edit embryos so that these children already have this mutation so that from the very beginning, they're resistant to infection. Well, the problem is, number one, this is a variant that appears in Northern Europe, so we don't really know how it would function in a population that is very different. Remember, genes are not isolated. They operate in context of other genes as well as environmental factors. And so what works in one way in a population in Northern Europe might not work the same way in a different population with a different kind of range of traits that are prevalent in that area. The second is that while this mutation may make you resistant to HIV, it can make you vulnerable to other diseases. This is the mixed blessing of many things. And we see this, for example, with sickle cell having some effect on resistance to malaria. If you don't have 2 copies of the sickle cell mutation, which will make you sick with sickle cell, but only have 1 copy, you actually have an advantage against malaria. Well, with CCR5, there's a strong hint in some of the literature that among the things you may become vulnerable to if you have this particular variant is certain kinds of viral infections and influenzas, which of course in a place like China is particularly problematic because we've seen a number of avian flu viruses that have then mutated into human use, into human application and infection coming from certain parts of Asia. So it's one of the last things you want to make people more vulnerable to, especially when there's no real purpose to it. I was a member of the organizing committee for the summit that was now being held in Hong Kong, the weekend of Thanksgiving, and on the night before the summit, we now knew that JK was planning to discuss the fact that he had brought these twins to term and that they now were living someplace in China, unknown where, with health status unknown, names not being revealed in order to protect their privacy, but also making it impossible to know how to check what he had done. We had very little information about who had collaborated with him, which IVF clinic had worked with him. He wasn't an IVF doctor. And yet, at the same time, that we had serious doubts about what he had done and tremendous doubts about the way in which he seemed to think he had met any of the kind of conditions that we had recommended from the national academies or anybody else's purported code. He seems incredibly sincere. I mean, this is the thing that's strange about him. He seems truly sincere about wanting to somehow save these kids from HIV, but he's also rather deluded because he thinks he's done something good and that it is worthy, and he thinks he's going to become celebrated like Bob Edwards, who was the father of IVF. I'm going to finish up by saying just 2 more things. When he finally did his presentation with hundreds of people from the press all crowded into the auditorium to watch, and he threw up some slides with some of the data that he says he had collected, one of the ironies is that he had, in fact, edited these embryos, but he had not actually achieved the edits that he wanted. So there was no way to think that these girls actually did have any kind of HIV resistance. Instead, they had yet other variants of CCR5 whose effects are as yet unknown. And each girl had different edits. So we really have no idea what effect this may have. We hope that it'll be benign, but we just don't know. And the Chinese authorities have launched an investigation, which is still ongoing. Only some of its results now made public. The second thing is that it has spurred activity globally to try and figure out how to constrain what we now tend to call rogue science in this area. Nobody can absolutely guarantee that you'll never have a rogue scientist. And certainly he seems to have evaded some of the Chinese domestic regulatory provisions himself. But there are now 2 major international efforts. One is at the World Health Organization, which had its first meeting in Geneva 2 weeks ago, which is looking at options for governance. We have some countries that have already banned this, other countries that have not banned it but have put obstacles in the way. That would be the United States, and other countries that haven't even thought about it yet. And so one of the questions is, what do we want countries to do and how do we want them to implement it, and whether or not countries should try to come to agreement. And there's a second effort that has not yet started, which has to do with collaboration among academies of science and medicine across the world to try and flesh out at a technical level what is the kind of information you would need to have from laboratory experiments, from animal experiments, in order to know whether something really is safe and effective, whether it does what you want it to do, doesn't do what you don't want it to do, is stable across generations. Because until you have that information, no regulator in any country is in a position to make a decision about whether or not this could proceed purely on a technical basis, putting aside the public policy discussion. So where we are right now is hoping that nobody else does what JK did himself. Thank you.
Thank you. I'm not sure if that was a positive ending or negative, but I'm sure we're going to push Alta to say more what she really thinks about this later in the discussion. But I think we forgot an important actor in all these affairs, the public, the citizens, and what do they think and why they think that way. And what does it tell us about communication, about these complex issues that revolve around new technology that have not only scientific but also ethical, legal, and social implications. So Dietram, 5 minutes to solve this conundrum, and then we want to have enough time for questions among the audiences and obviously among the panelists as well.
Thank you, Dominique. And thank you, Alta, for normally my students tell me that I end on the most depressing notes for my lectures, so you already did that, so I can only be more optimistic in comparison to that. I'm a faculty member in Life Sciences Communication, so the work that we do within the College of Ag and Life Sciences is to figure out how most of us make sense of technologies just like CRISPR that arrive unbelievably fast, that are highly complex, that most of us don't understand, but we're asked to make policy decisions and very often personal decisions. And as many of the previous speakers, including Pilar, have said, these are not new issues. Right? In 2003, Alan Leshner, who was at the time the CEO of the American Association for the Advancement of Science, the largest generalist science organization that we have, turned to science and said, look, you guys need to stop communicating the science to people, and you need to engage in a much broader, what he called bidirectional dialogue about science. And typically this is where when scientists quote him, that's why they stop, because the next sentence is actually even more important, but it is also more difficult. And he said, not just about the promise of science, but about the perils and the pitfalls. And I think we heard about a lot of them here, and Alta's discussion of the Chinese twins are a perfect example, because that catapulted the issue of CRISPR from an obscure discussion, or maybe not obscure, that's unfair, but certainly not a discussion among 320 million Americans, to something that a lot of people started paying attention to. And certainly also the report that we did. And I think in many ways what's really important is that to understand that CRISPR is a perfect example of what some people have called a wicked problem. What do they mean by that? They mean a problem that doesn't just have technical solutions that science can solve, right? We're figuring out how to do it, and once it's safe, we go ahead, and then the world is going to be a better place. It will be in some respects when it comes to curing sickle cell. We saw the technical complexities, and those will be sorted out. And the committee that Alta co-chaired and that I was on, I think even while we were meeting, literally the safety, the technical safety of the technology of CRISPR improved. We could almost see it in front of our eyes, right? So we will solve the technical problems just fine. The question are the ethical, the societal, and the political problems that are going to arise from it. And I just want to talk about a few of them that I think will trigger larger political discussions and really pose challenges in a media and information system where we have fewer science sections of local newspapers than ever before, where lots of local communities are news deserts, no longer have local papers, where most science writers that we have in this country are now freelancers. The vast majority of the National Association for Science Writers are actually freelance journalists because they're no longer permanently employed by news organizations, meaning the infrastructure of the people who used to translate complex science for us and told us why it mattered for our daily lives is deteriorating. So let me, just a couple of issues. One, for instance, if you've never heard his name, it's worth looking him up. His name is Ed Yu. He's an FBI special agent who looks into issues of hacking. And what the FBI is increasingly concerned about is not data hacking at hospitals for patient data like we usually understand patient data, or even financial data, but data hacking for DNA data. Why is that? Well, because whoever has the largest amount of data on the largest numbers of patients, and we see movements within China, for instance, to sequence the genome of newly born babies. It's a big data problem ultimately, right? What I would like to have is similar to what Google does. And what Google does was 23andMe, which was, of course, created by Sergey Brin's ex-wife. So collecting as much genetic data as possible and pairing that with two things. One is behavioral and other informational data, the stuff that all of us produce with our mobile devices on a daily basis, meaning Google knows, and so do most other tech firms, where I've been at any given moment, how much time I've spent in potentially lead-contaminated buildings, when I searched for that pain in my side, when I was in the hospital, what kind of treatments I got, what my Google Health data, my Apple Watch, and my phone, and so on. All these behavioral data matched up with DNA data, matched up with artificial intelligence to make sense of all of these patterns. Right? We know that Google already, that DeepMind AI, so this is Google's AI arm, is much better, has a 95% certainty at predicting your gender from a picture of your retina, for instance. That's something that most ophthalmologists can't do. So, and the funny thing is, or the depressing thing is that DeepMind actually doesn't know what the algorithm does. They just know that it works, but they don't know how the algorithm does it. Now, why is that particularly relevant for DNA? And it's partly relevant for DNA because, of course, there are lots of chunks of genome that we don't understand yet. We know that if we want to make edits, for instance, for intelligence, we'll have to edit hundreds of genes and combinations of genes, and we're just so far away scientifically from them. But there's certainly the quest for the Holy Grail to combine those different data sources and use AI in the process to do those things. So, looping us back, this is exactly why we need these broader societal discussions. Because do we want to go there? Do we want to make these edits if they're possible? And very often the response from the scientific community, and I look at my colleagues here, is we shouldn't have these discussions yet. They're unrealistic because we cannot do them and maybe we will never be able to do them. The counter argument to that is once the technology is ready to be deployed, it's too late to have those discussions. The technology will be deployed somewhere, and I think the Chinese babies showed this. Maybe that's rogue science, but somewhere among 7.3 billion people in this world in 200 plus countries, somebody will deploy technologies that are possible to deploy. And unless we have these broader societal debates in an infrastructure that's less and less prepared to have this debate, including the US, I think we're going to have real problems. So I'll leave it at that, and hopefully I was just a little bit more depressing than Alta than at the end. Thank you so much.
Please, the panelists, if you can join Dietram here on the podium. I mean, we will leave, I think we will keep the moderated discussion brief, because I think, as you all said, what's important to have, a discussion with the audience. So very brief, 15 minutes. And can we take that slide? I don't know how to do this. So I'm going to ask general questions, hopefully to stimulate the debate, and I hope you can all answer to the best of your abilities. What I think is great in this panel is like you all coming from very different perspectives, and hopefully we can combine those perspectives in something that will be useful from all of us. And I think that's what the idea is for those wicked problems that Dietram was mentioning. We can all have a say in what's important to take into account, and potentially compromise in a way to move forward, right? And so if we look at CRISPR and related technology, do you think there's dangers of overpromising or underpromising on the technology? How do we actually reach that compromise, and who should make those decisions? That's easier.
Sounds like it's a question for Kris.
Kris, are we overpromising?
I mean, I think we're pretty realistic about some of the hurdles that we have to get through to get into patients. This area is actually quite financially filled with all sorts of intellectual property and incentive to move things quickly to the bedside. So, certainly we have a lot of basic science being done, just trying to understand how specific can we get these editors, how can we get them to the right cells, et cetera, that I talked about. But as soon as some of that understanding is published, there are teams of engineers that are working in labs throughout the country and internationally, such that they could be packaged to say, fix the sickle cell mutation or fix cystic fibrosis mutation. There's a pretty strong infrastructure of patient groups in rare disorders in particular that are ready as soon as the technology comes on board. The basic science is there to show that you can do these types of edits to aggressively test them in patients. And so, some of these cases, scientists have direct contact with. Certainly, we've met some patients, say, with pediatric cancer that, as a human being and as our trainees see sometimes at conferences, they're very compelling stories. And so I think there's some sort of regulation there just internally not to overpromise. You don't want to say you're going to cure this child's cancer within 2 years when it's a tall order. So I think there's a natural check there. On the other hand, there is some level of description of what it can do such that you can get the sufficient resources to try to get there as quickly as possible as well. So those are maybe some insights into the balance that scientists have to make when they talk about this technology.
Pilar?
So I just want to say that in the 1990s, when we were doing so-called gene therapy, there was a lot of irrational exuberance in the scientific community, which people actually acknowledged. So what happened was maybe we moved some gene therapy attempts into human trials too quickly. One person was killed as a result of a trial. A couple of people probably got cancer as a result of being in gene therapy trials. It caused a lot of self-examination in the scientific community. People really actually were acknowledging that maybe they had moved too quickly, maybe they were believing their own hype a little bit too much. And also people recognized that because of these bad events, the whole field really slowed down and maybe even overcorrected and became overcautious for a while. So I'm hoping personally that it's a lot of the same people actually, or at least part of the same community. And I think that community, many people in that community did learn an important lesson in the 1990s. I think the other thing I would say, and this goes back to my justice point earlier, I was really excited in many ways that the NIH is really focusing on blood diseases as a first area of study for somatic cell gene editing, and there are technical reasons to do that. But one of the reasons they wanted to do this, and I know from the people at NIH who talked about this, is because sickle cell is one of their big target conditions to try and ameliorate or even cure. So sickle cell is it's a disease that hasn't been given the kinds of research resources that maybe it should have been given historically. So there are many studies on this. It's also a condition that affects primarily people of certain descent from certain parts of Africa and certain parts of the Middle East and around the Mediterranean. So that's all great that NIH is focusing on this, but we also need to figure out how people are actually going to get it, right? Because another kind of overpromising is developing a therapy that actually the people it's designed for can't really get because their insurance won't pay for it. So I just have to say that sickle cell disease right now, less than half of the adults who have sickle cell disease actually get the guideline recommended small molecule drug treatment that they should be getting. And then when when their doctors prescribe it, they often have a hard time getting insurance to pay for it, even though it's the guideline recommended treatment. So among the other things we need to figure out are the financial models to make it possible for people to get this therapy when and if it becomes available.
So, Alta, can you actually comment on the overpromising versus compromise?
Yeah, I think there's two different kinds of overpromising, to go back to the earlier topic, although they're all very much linked. One is the overpromising that comes from portions of the scientific community and the business community about how quickly we're going to be able to move to the point where we have an approved and marketed therapy. Now, we saw this with embryonic stem cell research, but a lot of it was because there was such a strong political backlash to that particular technology from some parts of the country, that there was tremendous pressure being brought upon the scientific community from Congress, and there were pressures to reduce or eliminate funding, to criminalize certain aspects of the research. And so one of the effects of that was to push back and make statements at hearings and in senators' offices privately that were probably way over optimistic as to what the timeline would be. And that is a natural thing. And for companies that are investing, of course, there's always the intrinsic conflict of interest that comes from wanting to encourage people to be excited about your company and its prospects within the limits of law. So that's one kind of overpromising that we've seen, and it happens particularly when you have a novel technology that has a lot of political opposition, so you're trying to push back against it. And I fear that some aspects of genome editing may run into that. There's a different kind of overpromising, though, that I think is actually even bigger risk that we haven't touched on. Again, I'm going to go back to stem cells. One of them is flim-flam, so-called clinics that advertise all kinds of purported therapies using the magic words of whatever is the new thing. Could be stem cells, could be nanotechnology, now it's going to be gene editing. And they suck people in who have a whole range of conditions, and they snow them with all kinds of technical language and give them who knows what. We have it going on right here in Wisconsin in Oconomowoc, and we have it going on particularly in the United States in Florida, Texas, California, and it's certainly been going on globally. It actually started mostly in places like Ukraine. The FDA is finally cracking down on this, but that is an even bigger risk because there's something really exciting about these new technologies, magic words, and people who've got chronic conditions that don't have good treatments. So it can be a variety of orthopedic problems, neurological problems, whatever, will often be willing to try almost anything that somebody says is going to work. And we don't have the enforcement capacity to go after every one of these places. So one of my fears is that the next wave of this kind of flim-flam medical tourism is going to be genome editing.
Yeah, so this is like, so the hope that the technologies bring, and obviously we generate people that are taking advantage of it from financial reasons. But we haven't talked too much about people that would be strongly against the technology. Dietram, can you comment on how some beliefs, I'm thinking of religious beliefs, for example, may come into play for people that actually are against these kind of things?
Yeah, and I would start out by making a distinction, because I think very often when it comes to emerging science, our reaction is, well, there's public resistance, and eventually you're going to overcome that. And we parallels, and I think we heard this on the committee, Alta and I, people saying, well, it's just like IVF, right? At the beginning, people are really upset about it, and eventually they're going to come around and they get it. And this is and they'll come around to the way of thinking about it like scientists do. And I think this is what I was trying to set up with a point about wicked problems, right? That some of the questions surrounding CRISPR and its applications, potential applications, beyond therapy, and Pilar said this earlier, we're not just talking about therapy, we're talking about applications that may go well beyond that. We just went through a USC and other school admission scandals where people have done things that are all out illegal to get their children into a private school. We've always seen selection in terms of who people had decided to have children with. People pay for Kaplan tests. People get Adderall prescriptions for their children. It's not unreasonable to say the next step is, why would you not edit the genome if that was possible for your child in ways that would increase the likelihood of your child doing well? Anybody who thinks that that's not going to be a motivation, I'm not saying it's going to happen, but that's not going to be a motivation, I think I would call us fairly naive. So then the question is, So people have reasonable concerns about this, and reasonable concerns may be concerns that are based on faith. The idea that we're doing research in some of those parallel previous technologies, that we're doing research in embryos. I happen to not be a person who shares those views, but that's irrelevant to the debate. That just shows that there are different viewpoints to that. There are arguments about, and Pilar outlined some of them really nicely, about hyper-discrimination. We talked to groups on the committee who, part of the disabled community, and they said, we don't want to be fixed. There's nothing wrong with us, a hard of hearing community, for instance. So I'm glad you're having a cure for us, but I don't want to be cured by you as a doctor. So what if some, or even a majority of those communities tend to be, or just choose to have that condition edited, and then the small group doesn't? Are we going to have hyper-discrimination against that group? Because why would you never get edited? Couldn't you afford to be edited? Why would you make that choice? And so you can think through a lot of very reasonable debates that need to be had. So I think I want to push back against the idea that there's opposition and we need to address that opposition so it goes away. And I would loop back to the idea we need to find a way to have these broader societal debates about which parts of what applications we think are useful, are societally smart, are ethically sound. And I think the idea of access, and this is again something that Pilar brought up, I think is a hugely important one. I grew up, I spent still the majority of my life in Europe, and I'm still in awe, and not in good awe, of a country that doesn't have preventive dental care for most of its citizens. So how are we going to, and the sickle cell example is a really good one, right? Or a really bad one, I mean, however you see it. But what is our mechanism for deploying therapies to people who right now don't have preventive dental care? And I don't have an answer to that, and I think it warrants a much broader debate. So my answer would be, there is some opposition that is clearly based on misperceptions. There is some opposition that's clearly based on not getting the science right, and our job is to make sure that the best available science inform those debates. But the outcome of those debates very often is a political question that needs to be decided in international regulatory bodies, that's WHO or whatever else, or in political fora, and I think that's really where the solution lies.
Yeah, I want to open the floor for question, but Jeff, can you at least, to be positive, give us a little idea of what do we do at UW-Madison, for example, to prevent our scientists from going rogue, and what kind of committee is actually making sure that we are not editing the germ of human embryos in the labs?
So here at UW, if you were going to do any kind of embryo research,
Right.
there's a committee, the Stem Cell Research Oversight Committee, who also oversees all the stem cell research on campus, would have to approve any kind of protocols that would be proposed on campus. And actually, if you did any gene editing in human embryos, it is prohibited to transfer them back to a uterus at this time. So that those works would be prohibited. And of course, the human subjects IRBs would also have to approve research where you're collecting eggs from patients or sperm from a donor as well too. So on campus, we already have some things in place. At least we're providing some oversight. We know what's going on, and I think you can prevent some of the rogue scientific experiments that have happened around the world.
If I may just, in addition, what you're talking about are the campus level controls. For people that are concerned about editing of embryos and resulting children, to do that, to transfer an embryo into a woman's uterus, you are now working in an area in which the Food and Drug Administration
Yes.
actually has jurisdiction. It doesn't sound like it should from its name, but it does. And Congress currently has a provision that says that the FDA is not allowed to even accept an application to start a clinical trial that would involve a pregnancy with edited embryos, let alone approve one. So for the moment in the U.S., anybody who tried to do that in any location, here or elsewhere, would actually be subject to civil and criminal penalties. So we have lots of guards against that. Somatic editing, however, can proceed, but again, with regulatory oversight from the federal government as well as our local oversight bodies.
All right, so why don't we open the floor for questions? I think we have some microphones around. Questions? I see one here, one. 2. Can you remember your number? To be fair, 3,
There's a couple over there.
Way on the left.
4.
There's actually a mic.
Okay. Oh, can you follow my number if possible? I just want to make fairness to people that ask it. All right. 5.
I had kind of a question about kind of how you'd profit on these things because, kind of to use an example like HeLa cells, where a corporation took something, well, not a corporation, a medical institution took something from a woman, her DNA, which I would say is her property intellectually and physically, and made a profit on it. So my question is this. If DNA, let's say, is a common resource, and a corporation edits it to make money off of it, at what point does it become that corporation's IP? And at what point is it something that you own by birthright? Or if we edit humans and we give those humans those edited genes, doesn't that mean a chunk of that person effectively belongs to a corporation?
Who wants to answer this? Pilar?
So I can answer that last part in particular because I teach patent law, and that's the area of law that might apply, except our patent law now explicitly excludes patents on human beings or human embryos. So the answer to your question, that last question, is no. It wouldn't turn that person into property. But the technology for making, CRISPR edits and particular kinds of CRISPR edited cells in the lab, those can be intellectual property, and they are intellectual property. And there already are patents on the technologies that are used in CRISPR. So.
Anybody wants to add anything? No? Let's go to number 2. I don't know who was asking. Here? Yeah.
Thank you for joining us tonight. My question is about adding mutations into the DNA. So in our current environment, there are many ways you can get different alleles, right? You can get a mutation in DNA simply by going outside and being in the sun. We are already introducing many mutations by pollution and by radioactivity in our environments. And I'm interested in how you can compare and contrast those mutations with intentional mutations implemented by CRISPR. Thank you.
Kris?
Sure. So many of those mutagenic processes aren't directed at a specific locus or a specific portion of the genome, whereas I think some of the technologies here are really going after, arguably in CRISPR, around a 20 base pair exact match of what you programmed the CRISPR to go in and tackle. And that 20 base pair type of number gives enough specificity in the human genome of 3 billion letters that you can go into one spot quite specifically. Of course, there's mutagenic processes that could happen during sperm development, egg development, and those are sometimes lead to very serious diseases. And part of the hope here is that if you can read those mutations that perhaps landed in a bad gene, we would have some technologies to go into those areas of the genome and change them and modify them such that you ameliorate some of the symptoms.
I had a question here.
Thank you all to the panel for the event tonight. My question kind of parallels a lot of the discussions about the community engagement. And if the panel could discuss a little bit about how the community is currently engaged at the various different levels, from basic research in labs, the oversight committees, the IRBs, up through the FDA, and how those mechanisms are organized and how they might be improved to kind of foster a better communication bidirectionally.
I'll take a first stab, and Alta can chime in as well, because I think, the report that we wrote, but I think every single report that came out of the academies on related issues, if that's gene drives, if that's GM crops, mitochondrial, there's one other that I'm forgetting right now, all actually now had whole chapters or at least large sections devoted to that very question. And they've landed in slightly different places, but I think in a very generally congruent spirit. And I would put a scale out there in terms of where the efforts are. And I think there's, on the one hand, and we had these discussions. This is also not the first technology where we've had these discussions. For nanotechnology, for a whole bunch of technologies that we rolled out, there has been a shift toward what the Obama White House, OSTP, under John Holden and others have called responsible innovation, or some other people have called real-time technology assessment. So this idea that as we're rolling out technologies, can we do an iteration over time where we're looking at its impact as it's being rolled out, and engage with various communities as it's being rolled out, and then feed that back into the research process, either in terms of directing the research in particular directions, or providing feedback on areas of research that we don't want to do. So let me come back to that idea of a range. On the one end of that range, and Vicki Colvin, who was at the time a toxicologist at Rice, she's now provost at Brown, she was on stage at an event that we organized, and she basically said, don't call it community engagement or public engagement if you're not willing to close your lab. So in other words, if a community comes to you and says, we don't want you to do this research here, you shouldn't do it. And I think we've had some run-ins with that spirit, both at Wisconsin, at Cambridge, at Berkeley. Insert, your stereotypical left-leaning community. And for almost every technology that has been pushed from the community to either regulate it to a point where it can be done, or to impose burdens of reporting that made it virtually impossible to do that. Is that good or bad? I'm not taking sides. I'm just saying that's one end of the scale. The other end of the scale is what I said earlier, and that is the idea that we're basically trying to manage public opinion. I think both ends of the scale are not ideal. I think and most of them fall somewhere in the middle. So the last part of the final part of your question, I think, is really the key one. Are we doing as good a job as we can in the current structure? One of the things that we said in the report, for instance, the Recombinant DNA Advisory Committee, the RAC, at the FDA has traditionally, for example, not included people who do this for a living, meaning who are trained at how do we best engage. And some people on the committee have been really good at this, but there has not been a systematic effort to staff the committee that way, or to have members of the committee that way. And one way of moving forward would be to build that expertise much more explicitly into it. But other things have been done at universities, and I think universities can play a unique role. I think this building obviously being a great example, but also when we look at why else do we have a unique role? Because we have higher levels of trust than virtually any other institution. That sounds weird, and intuitively a lot of people think that science has lost public trust. But we're actually, if you look at national science indicators, we're only second to the military in terms of the public trusting the institution, right? What has gone down is Congress, What has gone down is the press. What has gone down is industry even. But science enjoys, as a convener of conversation, I think, an unbelievably high level of trust. And I think using that for those kinds of conversations, and this comes back to my earlier point, conversations that are informed by the best available science, right? So where we're not going off on weird scientific mis or even disinformation, but at the same time have a broader debate that goes beyond the science. I think universities are uniquely suited for that kind of engagement. That's somewhere between those two anchors without hopefully hitting either one. That's my personal editorial comment.
So Alta, can you comment on that? Because I think that the crucial question is, what's the outcome? I mean, if we're having conversation that stay at the level of conversation, we will have some frustration from the citizens side.
For the mechanism.
So,
Well, I think it's important to maybe, because I'm a lawyer, I think in terms of structure. So let's start at the transnational level and then move down, and then you'll see what these conversations can and cannot do. So at the transnational level, there's a lot of interest in both the scientific and non-scientific communities in debating whether there should be a particular policy or a diversity of policies, if there should be a particular regulatory approach or not. And that is going to take place in a variety of ways. There is an organization that formed that's primarily Eurocentric called ARIGE, which is open to anybody, science and non-scientists, has ongoing online conversations and has had in-person meetings. There's also now been a call from the Summit Organizing Committee that we had in Hong Kong to create an international body that would function a little bit like the old Recombinant DNA Advisory Committee functioned here in the US. It would be a body that can be both a place where information is gathered about what's going on, who's doing what kinds of experiments, keeping registries, could do protocol reviews so that we could actually have in-depth review of a proposal before it even goes to a national regulator with advice about maybe you shouldn't do it or here's a better way to do it. And where it could also be a place to convene public debate, either among government leaders in individual countries or among representatives of interest groups. Then and then of course I mentioned the WHO committee is looking at international governance options that would include not only things like registries, but in theory could include treaties and conventions and such. At the national level, every country has its own way of deciding how the public will interact. In some countries, everything is allowed until it's forbidden. That's pretty much us. In other countries, everything is forbidden until it's particularly allowed. Those are very, very big distinctions, and that drives a lot of the policy, even if the public conversation was the same. Some countries, the public can control policy. Other countries, it's very centralized and the public has very little kind of influence. So that's going to vary from place to place. So how the public interacts is going to differ from country to country. Then within each country, there's going to be a set of regulatory options. And Dietram's already alluded to that. In the United States, we have the Food and Drug Administration as a regulator, regardless of where your funding comes, of what you can and cannot do that involves this kind of cellular manipulation and transplant into a person's body. We also have funders who can put controls on what you can do with their money, and the NIH is certainly the biggest actor on that, and they can have a profound influence on what's done. Doesn't maybe make things illegal. You can find alternate funding, but this is really very influential. And then at the institutional level, which Jeff had alluded to, even if everything is permitted in your country, the local oversight bodies will look very specifically at what you're planning to do with this experiment and these people and these materials and ask, not only is it legal, but does it satisfy our notion about what is ethical? Does everybody involved know what they've gotten into? Are you doing something that has risks that exceed anybody's notion about possible benefits? So you get multiple places in which there's going to be public participation, but it won't be the same in every country.
Yeah. Next question. Where were we?
Hi, I have a question about how we can hold scientists more accountable for communicating the perils of the work we do. We seem to function in a system where even the publication of negative results is not routinely done. And in the race for funding, how do we incentivize these communications?
All right, so how can we hold scientists more accountable? Who wants to take this?
I'll take a first step just because you talked about incentive systems and everything else. The National Academies of Sciences, Engineering, and Medicine is currently conducting a consensus study, and we will present our results, I think, at the end of May, early June. It deals with exactly that question, and that is, it started with a concern about science being, or the perception that maybe we are in the middle of a replication crisis, that that some of the results don't hold, and that some of that is driven by exactly what you're talking about, scientists overpromising or maybe doing p-hacking and harking and all these things, so hypothesizing after they know the results and so on. That report will speak without being able to say what it will say, but it will speak to those topics. Unfortunately, I can't tell you what we're going to recommend.
Come back for a conversation.
But I promise you that you will find answers that will directly address that question. The second part that I want to talk about, though, is both Dominique and I have been doing surveys of the scientific community in particular areas of expertise for a long time. So this is trying to figure out what the leading experts, based on their publication rates, their impact factors, their citation rates, how they think about the idea of public engagement. Some of the early work that Dominique did was published in Science, I think now more than 10 years ago, right? But since then, what we've looked at in particular is, are there generational differences between an older generation of scientists that maybe has lived their whole life within the ivory tower, both mentally and sometimes physically, and a younger generation that has a very different outlook on communicating and connecting with the public. And you see in the data very nicely that we're seeing a younger generation that's coming up who, for example, are not as closed off to sharing something that they think is really important for public decision-making or policy decision-making, even if it hasn't gone through peer review yet, and saying that's urgent. And I think a lot of the movements you're seeing around archive and pre-publication listservs or platforms really kind of reinforce that idea that they are seeking that broader public debate, and I think that's very, very encouraging. And I think we're seeing really a generational shift. And not to put Kris on the spot, but I think Kris is one of those perfect examples of somebody who is obviously a leading bench scientist, but who has also written one of the earliest pieces urging the scientific community to engage more broadly on the issue of CRISPR and seek those broader societal debates with colleagues at Arizona State and Harvard. You can speak to this better than I can. I don't.
Yeah, so Kris wanted to comment, and then
Yeah.
Alta also wanted to follow.
Sure. I think this is a very interesting question of what scientists will take one credit for and also be responsible for, right? So at one level, it seems ludicrous that the first, discoverers of CRISPR should be responsible for where their technology gets used everywhere in the world, and including, the Chinese experiment. That's not, I think, a reasonable way to think about accountability. But there's also types of questions that we delegate to, questions about ethics, and we certainly hope that scholars such as many here and others at various institutions chime in on and tell scientists, that's really a line you should not be crossing, or if you cross there, these are some types of experiments that really need additional layers of oversight. So that's one sense of thinking about accountability. And then I do think part of the infrastructure of how we do and move forward responsibly does involve scientists to be not just delegators of questions, but also to participate. So you will hear leading scientists in this field generally being worried about some applications as soon as they realize some of the directions of where it can go. Jennifer Doudna is one of these examples, one of the leaders in this field. And so she wrote a book. She started organizing some summits, called up some scholars and thought leaders and also policymakers, and started to initiate some of these conversations quite early. So, in some ways she felt accountable for how the technology gets circulated and distributed. And I think certainly that's one of the questions that I pose to my students in my courses, to think through creatively on how they would have, moved forward if they were in her shoes, and what types of questions would they ask if, as they leave the campus and do great things in science. So these aren't easy questions. I do think one of the unique things here at Wisconsin is that we have really a nice group of scholars that talk across disciplines, that are able to think creatively about some of these hard questions.
So this question about the ethics of where your science goes transcends just the human applications we've been talking about here. One of the areas of real concern has to do with engineering, bioengineering, that would make it even easier to create biological weapons. And one of the major efforts going on now to figure out how to discourage this development is being spearheaded by something called the Nuclear Threat Initiative, which has a biological program within it. And one of the things that's emerging from that group is the idea of essentially an ecosystem approach to these things. No one entity is going to be the one that can handle the problem. What you want to have funders put controls on what they will or will not fund. You want to have journal editors acting as monitors that whatever they're going to publish is coming from a lab that has met certain kinds of standards for guarding against misuse of their work, has met all of the local regulatory standards, has adhered to any professional codes that have been issued, that suppliers like DNA synthesis companies are given some kind of incentive to be wary about some of the requests that they get for materials before they simply send them out willy-nilly. That's a big problem in terms of business models, because those companies operate on a very thin profit margin, and so the goal is always to drive your prices down competitively. So we've got to figure out how to correct the market disincentives to this kind of thing. To create regulatory pathways that give preferential
treatment to those entities that have somehow met certain standards and been certified with like a seal of approval. Educational modules that are made available so that laboratory scientists at every level of training are aware of where there might be risks. If they do this, then it might be diverted to that. And it may be that we just want to make them aware of it. It may be that we want them to be more conscious of what controls exist. It may be that we want them to think about whether they shouldn't do it at all, or if they're going to publish, that they should publish in a way that restricts some aspects of it. So it's about looking across all of these different possibilities and then coordinating them to create a kind of an environment in which you have different controls that are actually kind of leveraging one another's power so that you get backstops and backstops.
I think we have time for one more question. Yeah, you were trying to reach my two more questions. Keep it brief. Keep brief the answers so we can let the two gentlemen ask their question. Go ahead. He was waiting for a long time for a question here. Sometimes it's hard, you're in the front, so you don't.
Following at that a little bit, I just have a question sort of on the pace of this research. So obviously editing one genotype can be linked to multiple different phenotypes. And if we were to wait to fully understand the ins and outs of every single gene, it would take a very long time, right? So how do we balance sort of, obviously we want to get these therapies in the clinic and into the clinical trials. So how do we sort of balance knowing the consequences and the potential effects of the research with just trying to push it into the clinic as soon as possible?
Pilar? Oh.
I think the level of understanding that's sufficient to go into the clinic is mostly, I think, submitted in terms of a preclinical data package to the FDA. And there's a back and forth process, and how much of mechanism of action, for instance, do you need to have in terms of gene function can vary. And I think there's quite a bit of negotiation there about the level of evidence that you need to get into first-in-man. And then that's just the tip of the iceberg. They need to go through three phases of clinical trials.
I mean, the fact is you balance the risks and the benefits. So if you've got an urgent need for something that will correct a serious condition, the level of evidence will be a little bit more flexible. If it's something that's really got alternative strategies, or it's problematic, it's uncomfortable, but it's not going to really reduce the quality of your life, then the evidence requirements will be far more stringent. So it's a benefit-risk balance.
And this is something that, it's not unique to CRISPR. Again, this is kind of the FDA's job. It is Congress's job, right? Congress has helped to set the policy that the FDA, and the law that the FDA uses to make these balancing calculations. And exactly this point that Alta just made, right? That things that are super serious medical conditions for which we don't have an existing therapy, the FDA can actually let those onto the market with slightly less evidence. So it's actually a kind of condition by condition determination. It's not just a global determination for all of CRISPR.
Dietram, very briefly, and then one last question.
Yeah, and I just want to, because I think we're looping back to the idea that the risks and benefits are largely technical in nature, and I think your question was broader than that, and that what are the long-term things. One of the issues that came up on the committee again and again, and John Evans, who was a member of the committee, has spoken about this publicly, is the idea of a slippery slope that he is very concerned about. Meaning that, yes, every single step that we edit, that we make in every application may by itself not be a problem, but as we're pushing this further and further, we may end up at a point where we don't want to be. If 20 years ago I had told any person in this room that you will carry around a device that at any given moment will tell corporations, the government, where you are, record everything that you say, will have its microphone on, every email that you write will be read by Google and Gmail and whatever else, there would have been a civil war. And that you're going to carry this at all times, right? Wherever you are, you will have it with you. It would have been a civil war. But then we had cell phones, and it was really cool that we could speak without being on a wire. Then we realized that we could have email, and then we realized there were maps and there was location services. So we went step by step into a decision that we have never made as a big decision but we made a lot of little ones that got us to a point that we would have actively rejected 20 years ago. And that's kind of the same tricky part, right? That every application by itself may be perfectly fine in terms of technical risks and benefit calculations, but cumulatively, where we are 20 years from now, or where certain groups of the population are not 20 years from now, maybe a different story. And I think that's an argument worth considering, at least. Not that I'm promoting it, but I think it's an argument that's worth considering.
So we have 1 minute left, so you get to close the debate.
Pilar and Alta talked on the topic of consent for germline editing, and I was wondering, is that a hurdle that you ever see the field moving past, is addressing that, and if so, who do you think the table would be in that conversation about whether or not that moves forward past that hurdle?
Consent.
So, first of all, of course, if you're a person whose embryo might be edited, you will consent, right? So the real issue is that the person who would be born and their whole lineage, next generation would not consent. And as I said, frankly, everything we do affects future generations, and they can't consent to any of it. And so the idea that it cannot possibly be the case that we can't do anything because future generations don't consent to it. Instead, we need different ethical rules to help us know what we can do, when we can do it, what kinds of risks we can impose on the future, what steps we should take to minimize those risks. So I think in many ways we've already gotten beyond that question. It's also the case that people in the future don't consent to be born with a very debilitating genetic disease, right? And so if you do nothing, if you don't move forward with the technology, you're also making choices that affect those people. We just can't help that. Our choices will affect them. And so the question is, what ethical framework do we apply to make better choices rather than worse ones?
That's a great way to conclude this panel. Please join me in thanking our volunteers, and thank you to the audience.